So today we have the opportunity of interviewing Mr. Sanjay Yadav, which is the general manager of Meril Life Science. Is that correct?
Yes.
Well, thank you. Thank you for coming and welcome to Madrid to CSC 2018.
Thank you.
Mr. Yadav, could you give us a very brief overview of your company, which is Meril Life Science and is distributed in Spain through Palix during the last 5 years, I think, if I'm correct?
Yes.
Could you give us a very brief overview of the company so we can locate you?
Well, Meril is a part of a large Indian business group called as Bilakia Group, and we started our healthcare journey in the year 2007. So it's almost 11 years of journey in healthcare. And from the time we started with cardiovascular, but now we have extended in orthopedics, endosurgery, in vitro diagnostic and ENT, and there are several more business verticals to come from. And we can proudly say that we are one of the fastest growing medtech company from Asia.
Wonderful. From the intervention and basically from the cardiological intervention point of view, you have a very interesting new product that has been recently redone, should I say, which is the new bioabsorbable stent, coronary stent, and that is what I think we should focus in the next minutes. And regarding the stent, I would like first for you to review the main features that differentiate it from previous stent, both your first generation and the first generation of other type of bioabsorbable scaffolds.
So after the discovery of metallic stents, a drug-eluting stent has been one of the biggest discovery. But moving on in the segment of interventional cardiology, the next big thing which has been discussed is about bioresorbable scaffold, and we can proudly say that Meril is pursuing the project of bioresorbable scaffold very actively. So in terms of bioresorbable scaffold, right now there is an aura about where this technology is going to head, considering the challenges which we have seen in the first generation scaffold. So in terms of Meril's development of bioresorbable scaffold, considering first generation, which were having a strut thickness of 150 microns, one of the biggest change that we bring with the second generation scaffold, we reduce the strut thickness to 100 microns. So the name of our device is MeRes 100, which is Meril's resorbable scaffold with 100 microns. And in terms of characteristic and features for the device, the differentiating parameters apart from the thinner struts, which are now very close to the metallic drug-eluting stents, we also have a very unique hybrid cell design, wherein we have closed cell design on the edges, open cell design in the mid segment. So closed cell design always helps in maintaining the radial strength, while open cell designs allows better navigability of the device. In terms of drug and the polymer coat, we are still using PDLLA as the polymer coating, along with the most established and one of the most proven drug, which is sirolimus, with the drug dosage of 1.25 micrograms per millimeter square. So while I talk about the differentiating strut thickness, differentiating design, we also have in terms of radio opacity, 3 triaxial markers on each end. In fact, each radio opaque marker is a couplet on fluoroscopy which appears as one marker. So we have 3 markers on each end at 120 degrees equidistant from each other, and which enhances and gives very good radio opacity. And in certain situations without any imaging system, still we can consider with the help of these 3 radio opaque marker precise positioning and visibility of the scaffold.
Well, as you know, of course, strut thickness has a very large influence on radial force, radial deformation. How have you managed to achieve a reasonable radial compression resistance with a lower strut thickness?
Sure. So in terms of radial strength, you have asked a very valid question, that how we could reduce the strut thickness and still maintain the radial strength. As mentioned earlier, 100 micron strut thickness, and along with that hybrid cell design, it helps us achieve and maintain adequate radial strength for our scaffold.
You mentioned also deliverability. Can you delve a little bit more into the how you perceive in your company the deliverability capability of this stent as compared to others? Because I've seen you've introduced 40 millimeter length stent, which is really remarkable if you consider bioabsorbable scaffolds.
So deliverability definitely was one of the concern area in the first generation scaffold, and once again hybrid cell design with the closed cell on the edges. Majority of the scaffold consists of the open cell design, so it helps in the flexibility and navigability of the device. There are several bench tests which we have performed, as well as the cases which are done on human beings. We have seen that the device almost behaves like a metallic drug-eluting stents. With 100 micron strut thickness and a special crimping technique, the profile for 3 millimeter MeRes 100 is equivalent to 1.2 millimeter. If we compare this against the first generation scaffold, which had the crossing profile of 1.4 millimeter, while if we compare 1.2 millimeter of MeRes 100, which is almost equivalent to the second generation drug-eluting stents which are currently available in this scenario.
That's excellent news for bioabsorbable scaffolds.
Just one point on the same line, we have introduced 40 millimeter length, as you rightly mentioned. While the first generation, there were limitations in terms of diameter and lengths which were available. 40 millimeter length itself speaks about we'll only bring it when we are confident and we have enough backup and data to prove that this device is definitely deliverable like metallic drug-eluting stents.
Well, as I said, that's excellent news, but always we have to go to the gold standard and see how it behaves in human. What can you tell us about the clinical experience in human beings, and the first in man and the other studies that I know you have?
So we have
specifically considering the faults of the first generation, which is mainly stent thrombosis.
So we had enough evidence from the preclinical work which we did, and based on the success in preclinical work, we initiated the first in man. The name of the trial is MeRes 1, which had a patient population of 108 patients. Primary endpoints were MACE, major adverse cardiac events, in terms of TLR, TVR, as well as cardiac death. And in terms of secondary endpoints, we had procedural success as well as stent thrombosis as secondary endpoints. We have done this trial of MeRes 1 back home in 11 different sites, 11 different centers. And we can proudly say that in recently concluded TCT 2018, we presented 2.5-year follow-up data of MERES 1 trial, and we had excellent outcome from this trial. So in terms of primary endpoint, at 2.5-year follow-up, we had the MACE rate of 1.87%. And this MACE also we had no MACE, no instant thrombosis, and instant thrombosis was one of the preliminary concern in the first generation. So we have successfully demonstrated from MERES 1 that we don't have any stent thrombosis at 2.5-year follow-up. Within this follow-up, we have also angiographic follow-up, including the IVUS, QCA, and CT angiography results. The core lab, we had the clinical core lab of CRC in Sao Paulo, Brazil, while in terms of imaging core lab, it was done in cardialysis in Rotterdam. And the results are already proven in FIM trial with 2.5-year follow-up.
Well, just to thank you very much. Just to end up the interview, such a dynamic company as yours with a big interest in research and development, what are there any products that would you like to highlight that you have in the pipeline right now? Please. A short view from your company?
So considering our discussion on bioresorbable scaffold, we are quite confident as a company about this technology, and we are only extending the bioresorbable scaffold beyond coronary, taking it into peripheral intervention. But as a company in interventional cardiology, one of the prime project for us at Meril is our TAVI project. And we have a fantastic balloon expandable technology which is coming through. The name of the device is MyVal, and for MyVal also we successfully presented the initial FIM results at TCT this year, and it is just a matter of time before we come with long-term robust data. But in acute phase, 6 months follow-up, first in man, 30 patients, the device is looking extremely promising. So in terms of TAVI, MyVal is the next big project for we as a company.
Well, thank you very much, Mr. Sanjav. Congratulations for your results, and this will be the end of the interview. Thank you.
Thank you. Thank you very much.